All right, Dr. Whitaker is a postdoc that's in our department. He's been around here before, but he's doing some really fun work, kind of postdoc with soil chemistry, and he bounces around a few things. So with that, Andrew?
All right. Hi, everyone. Like Dr. Arnold said, my name is Andrew Whitaker. I'm a postdoc here at Oklahoma State University, and I work primarily kind of part-time in the soil chemistry lab, as well as the soil testing lab and method development here at OSU. There we go. And today I'm going to talk about soil organic matter and its many roles and applications, or pretty much many roles in agriculture. All right. Just want to mention, so this will primarily be a review of organic matter, not a lot of research. And so just kind of a general overview of soil organic matter. So today, kind of the outline and like where we're going in our presentation. So I'll briefly begin with what actually is soil organic matter. I'll also talk about soil organic matter formation, as well as kind of get into a little bit of organic matter functional group chemistry. So functional group chemistry is really important when it comes to these intrinsic kind of soil organic matter properties that everybody, that a lot of people are familiar with, such as like water holding capacity and infiltration and pollutant transport in the environment. So we'll briefly talk about that. And then we'll start to focus on some of the current research that we're doing in, or at the Oklahoma State Soil Chemistry Lab. And then the last thing that I want to talk about is soil priming. So what exactly is soil priming? And that'll help us set everybody up for the next presentation given by Dr. Yilling. So there won't be too many slides with a lot of words on it, but the first one, there's a lot of words. So what exactly is soil organic matter? So organic matter is an intermediate phase of carbon that's somewhere in between photosynthetically fixed carbon that's in plants, animals, and microbes, as well as completely oxidized CO2. So it's been studied actually for quite a long while, so around 2 centuries. So it's very important, it's very studied. There's tons and tons of research out that's on soil organic matter. And SOM consists of many different forms that have varying turnover rates. So some organic matter lasts in the soil for a really long time, up to thousands of years. Some of it is more simple and doesn't last as long and can be degraded by microbes, so over a span of 1 to 50 years. So organic matter in soils is very component because it gives a lot of the soil's properties. So things like cation exchange capacity, pesticide retention, nutrient availability, a lot of these things in the soil come from soil organic matter. So for starters, so how exactly does organic matter in soil form? So when we look at organic matter formation, I'm going to kind of, I'll rotate here. But so the first thing you got to have is, plant input. So that's the general kind of model. So you have plant input, your plants grow, your root mass grows, and then over time, your biomass is going to degrade into smaller particles. These smaller particles are going to fragment and go into different groups. So they're going to go into particulate organic matter, which is just abbreviated as POM, and then even smaller sections will be further broken down into mineral-associated organic matter. Some of the key things that you need to know about these fractions are that particulate organic matter usually lasts for a less or a lower amount of time in the soil. So particulate organic matter can last on average from 1 to 50 years in the soil, sometimes less, sometimes a little more. It's usually a lighter fraction because it generally just takes into account just organic matter. Organic matter is very fluffy, light, has a low density. But the compounds that are in the POM fraction are usually more complex. So they're bigger particles. They have a lot of different organic functional groups. And generally, they have a higher carbon to nitrogen ratio, whereas the mineral-associated organic matter It can last up to 10,000 years plus or minus a few. It's a heavier compound, so what that means is usually mineral-associated organic matter is associated with minerals that are more dense, and so this fraction will be have a higher density. However, though, the organic matter that's in the mineral-associated fraction is usually less complex, so they're smaller particle sizes. And then, as well as the mineral-associated organic matter also has usually more nitrogen in it, so the C/N ratio will be lower for this fraction. And so when we look further into the components that make up mineral-associated organic matter in POM, some that are probably a lot of you are familiar with, if you've heard things like lignin, cellulose, hemicellulose. So these come from plant litter. They're very complex organic matter components. They're very big fractions. They have lots of different types of functional groups, which we'll get into a little bit further down the road. So other molecular components that make up organic matter are things like microbial secretions and necromass. So microbial necromass is when the microbes die, they can then be bound into the soil and converted into organic matter. There are other things too, such as plant secretions, as well as dissolved organic matter. So all of these are a lot smaller size fractions, and they can be converted into both POM or mineral-associated organic matter. Some of the plant exudates are organic acids, so they're very small carbon components, and they can control a lot of different chemistry, and they have a lot of functions in the soil, which you'll learn more about plant secretions in the next episode of this crop school, so the next presentation. Other things that are associated with mineral organic matter are amino acids, and so all of these are very important components that make up organic matter. So the next thing I want to talk about that's really important when it comes to organic matter are the organic functional groups. So you can further break down your molecular components that comprise organic matter into simpler kind of subunits, which are the functional groups. The functional groups control a lot of different chemical properties in the soil, and they're very important. And whether it's related to pollutant transport in the soil or nitrogen mineralization. So the reason organic matter can supply your soil with nitrogen or sulfur or phosphorus is because of all the different functional groups that make up organic matter. So there's everything from alkanes that are more like hydrocarbon associated, all the way to carboxyl groups, which are primarily acidic functional groups. There's also things like phenols that are closely related to lignins and tannins, as well as different functional groups that have phosphorus in it, like phytic acid groups, as well as sulfur functional groups. So like I said earlier, these functional groups give rise to many different chemical properties that are really important in the soil. So why should you care about these functional groups? Why do we care? Why do we research these functional groups? So these functional groups give rise to many organic matter properties, some of them being kind of typical water holding capacity, which I think is thought about quite a bit, aggregate formation, so usually the more organic matter you have, the better your aggregates, whether they're micro or meso or macro aggregate formation is going to be better in soils that have more organic matter. Water infiltration is another one, your pH buffer capacity, pollutant fate and transport, whether it is hydrocarbon spills in your soil or whether you're spraying pesticides. So soils that have more organic matter can usually absorb or bind more pollutants. And the last thing we'll talk about is nutrient cycling. So we're going to briefly go into each one of these 6 organic matter properties. There's a lot more properties, but these are the 6 that are kind of I'll be focusing on today. So for the first one that I'm going to talk about is water holding capacity. There's numerous research articles that show that when your organic matter content in your soil increases, your available water capacity or your available water holding capacity in your soil will increase. So a general rule of thumb with this research, this research took a lot of actual soil samples. They measured organic matter percent, and they also measured water holding capacity in many different soils. They used that data to build a model, and from that model, you get this graph where you have on your x-axis your organic matter percent, and then your y is available water holding capacity in liters. And what the model shows is that when you go up in 1% organic matter gain in your soil, on average you can have around 11,000 liters of water stored. So if you go from 1% to 2%, around 11,000 liters of water more can be stored in your soil, and which that equates to around, so 1 gram of soil organic matter in your soil can hold up to 1.5 grams of water. So this is where a lot of your water holding capacity in your soil originates from. The next thing I'll talk about is aggregate formation. So a lot of research shows that as your organic matter content in your soil increases, you're going to have better aggregate formation. And so this occurs by the organic matter actually sticks to your soil particles, and then over time those particles in the organic matter can become larger and form bigger and bigger aggregates. And these aggregates can lead to many different properties, such as better water infiltration, which I'll talk about next. But just to show you what the aggregate, like the initial aggregate formation looks like. So this is a picture, a zoomed in picture of a biotite, so a clay mineral. This bar right here is 1 micron, so it's zoomed in a lot. And then what you can see is between these mineral pieces, you have organic matter that it's binding these smaller mineral pieces together, forming larger aggregates. So that's what it kind of looks like on a more of a molecular scale. And then when on a bigger scale, what you can see is that soils with this organic matter, or more organic matter, are going to have more larger, particles and aggregates. Not only that, but the particles and aggregates will be more stable. So, let's see, that's right. Okay, so when your organic matter increases and your aggregate formation increases and you have larger aggregate sizes, you also get better water infiltration rates. So in this study here, what it shows is that when your organic carbon in your soil increases, on your x-axis, you're going to have a faster rate of flow of rainfall into your soil. So higher carbon is going to lead to this better aggregate formation that eventually, over time, will lead to better infiltration rates of water. Okay, so for me, like I said earlier, I'm a soil chemist, so I had to get the soil physics and water infiltration and water holding capacity. So I talked about that first, so now I can focus on the fun stuff that's chemistry, right? So the next few slides are going to be more kind of chemistry focused and how our organic matter functional groups give rise to certain properties of the soil. So the first thing that I'll talk about is the soil's ability to buffer pH. So it's a buffer capacity. What research generally shows is that in pH ranges between 4 and 7 and 8 to 10, you have these functional groups that are weak acid carboxylic acids as well as phenolic functional groups. And these different functional groups will buffer the soil's pH. So what they do is when you have a pH change, these organic acids will deprotonate and leading to a hydrogen ion, so acidic ion in solution. So when you're applying your lime to soils that have low pH, if your soil has quite a bit of organic matter, you're going to have to apply more lime. So if you have a sandy soil, not a lot of organic matter, your pH is 4, you're going to have to apply less than if your sand had a lot of organic matter in. And the reason is because the ability of organic matter to buffer the pH. For this very same property, so when functional groups deprotonate, they become negative charged. Those negative charged functional groups can react with contaminants or hydrocarbons or pesticides in your soil. So I don't know if everybody's heard, but it's pretty popular in kind of environmental and soil chemistry, but remediation of PFAS. So these are perfluoroacting sulfonic acid. So it's a compound that usually has 8 carbons in it, and it's saturated with these fluorine ions. Unfortunately, it's very thermally stable in soils, and it's hard to break down. So microbes don't really like to break it down into CO2. And so there's lots of research going into looking at organic matter and how organic matter can react with these compounds. And the general consensus is that, once again, so as your organic carbon percent of your soil goes up, you're going to have a higher amount of absorbed contaminant. So, which is a really good thing, because you don't want this stuff in your drinking water or going to your crops or to your animals. So generally with soils that have higher amounts of organic matter, they're going to be able to absorb or take up and bind this potentially toxic contaminant. This also applies to herbicides as well, hydrocarbon spills. So the more organic matter you have in your soil, the better off you'll be for absorbing and immobilizing contaminants like this. This also works the same way for heavy metals. So if there's heavy metals in the soil, organic matter will buffer that and control the movement of heavy metals in your soil. The last thing I'm going to talk about before we get into kind of current research that we're doing at Oklahoma State University is talking about nutrient cycling and its role from organic matter's perspective. So there's negative charges, like I've said, in your organic matter groups. These functional groups, they deprotonate, they become negative. Negative charge functional groups like this oxygen, and when this one deprotonates right here, will bind metals in soil solution. So where this metal reacts with this negative charge, it's bound up, it releases a proton, but some of the nutrients that can be absorbed are things like calcium, magnesium, and ammonium. So if your organic matter percent in your soil is increasing, usually your CEC or your cation exchange capacity also increases. Another thing that we kind of think about in terms of organic matter and nutrient cycling is mineralization. So mineralization of nitrogen or sulfur or phosphorus in organic matter is a microbial process that occurs in the soil. And then what that does is over time, when the organic matter is mineralized, that will lead to actual fertilizers. So there'll be functional groups that have ammonium and nitrate in it. Microbes can go in there, they can mineralize the organic matter, and that will lead to ammonium and nitrate, things like phosphate and sulfate that strictly come from, that can come from the organic matter. So now I want to talk a little bit about the current kind of organic matter research that we're doing at Oklahoma State. So first, I want to talk about kind of classification of organic matter. There's kind of two methods. One's an older method, one's a newer. We're doing a lot of research with classifying organic matter with a new method. The old school kind of method is called humidification. A lot of you probably have heard of that, and hopefully some of you have heard of the fractions POM and MAOM. So dividing the soil organic matter into these newer fractions is something that we've been working on, and I'll talk more about that in the next few slides. And then the role of mineralogy and organic carbon. So I'm a soil chemist, mineralogist. I like looking at all the different clays and clay types in the soil. So I've been working a lot on that, doing a lot of different types of methods to identify if there's 2-to-1 or 1-to-1 clays, different types of iron oxides, to see how different types of clays interact with organic carbon. And so I really want to know if different ratios of these minerals can lead to different organic carbon sequestration rates. And the last thing that I'll talk about is soil organic matter and organic nitrogen mineralization rates. So what in the soil actually slows or speeds, increases this process. So for those of you unfamiliar with the humidification theory, so this is more the older school method, which the humidification theory primarily considers only plant debris inputs or plant litter input that goes into formation of organic matter. So how this starts is that basically your organic matter formation will start with a decay of simple organic compounds like sugars and amino acids first, so the microbes eat those first, and then what you're left with eventually is things that are more complex like lignin, and that is your mature organic matter. And the soil organic matter, though, is fractionated with harsh bases and acids. So this method is over 100 years old, and we've learned a lot more since this method came out. But generally, back in the day, you would fractionate your soils, you would extract your organic matter with the harsh acids and bases, and that would lead to usually 3 different organic fractions. So your humin fraction, humic acid, and fulvic acid. So you probably heard a lot about humic and fulvic acids. So this method was the original one that separated your organic matter into these 3 distinct groups. However, though, so this is actually some of the research that we're working on now. We've sampled a lot of different soils across Oklahoma, and for each kind of soil location, we have a managed, so a tilled location, and we also have a grassland location on the same soil series. So we have everything from like a Teller series, which is down in Perkins, Pulaski, which is west of Stillwater, and we have, Hollister soil series, which is down in Altus. And so what we're seeing is that in these managed soils, so where they've been tilled, there's little to no lignin. So the Teller series, there's little to no lignin when compared to the grassland soils. And that's pretty common throughout all of these series. So for Stillwater and the Kirkland, there's little in the cultivated wheat, but when you get into the sod or grass, there's more lignin. So that kind of, pulls up the question of, from earlier, so if lignin is mature organic matter that's really hard to break down, then why are we seeing little to no lignin in systems that are plowed? So that kind of throws out that method. That method being that lignin components are more stable, which is not so. So that brings us to the new kind of method, how we classify organic matter. So this new method uses, it's still a wet chemistry method, so we use neutral or non-harsh extractants, or we can physically sonicate the soils using high-frequency sound waves to disperse your soil. So instead of using these harsh acids and bases, we can use either high-frequency sound waves, or we can use, like I said, non-harsh extractants. And so what we do, we shake them for a while or sonicate them for a while, And then we put them through a wet sieve that kind of breaks them down into different fractions and size fractions, that is. So when when it goes through the wet sieve, we consider everything particulate organic matter that's greater than 53 microns and less than 2,000 microns. And mineral-associated organic matter is everything that's smaller than 53 microns. This new model not only considers your plant inputs, but the good thing about the newer kind of model of fractionation and classification of organic matter is that it not only considers plant inputs, but it also considers other dissolved organic matter, and it considers microbial inputs as well. So we're now learning more, so we're including things that are not only plant debris, but we're also including plant exudates and microbial exudates and necromass. So all of that comes together to make these organic matter fractions. So in general, earlier I showed that the humidification theory kind of shows that lignin is your more stable compound, and we currently have research, not just us, but this is done, there's a lot of research done on this that shows that lignin is not very stable and actually has high, mineralization rates. And the interesting thing is when we consider the new methods of classification, they actually shows that the mineral-associated organic matter is the fraction that is generally more stable. So this is the fraction that is less complex. It's less complex. There's smaller subunits of organic matter, but they form really strong bonds with minerals in the soil. So even though they're smaller subunits of organic matter, they can form these really strong bonds with your minerals within the soil, and that kind of protects them from microbial degradation. The next thing that we're also currently working on in the lab is I'm working a lot on this kind of portion, but what geochemical properties control organic carbon sequestration throughout Oklahoma soils. So in general, the rule of thumb is that, if you remember back from soil chemistry, pH is your master variable, right? pH controls virtually everything within soils. And so in general, what research has shown is that depending on whatever your soil pH value is, different elements or minerals control your organic carbon sequestration in your soil. So at lower pHs, you're going to have sequestration dominated by aluminum and iron ions. And as the pH of your soil increases, other things, so SROs are just iron and aluminum oxides. And then when your pH even increases more to above 7, you start to see that calcium and your clays, so your aluminosilicate clays, start to play a major role in this carbon sequestration. And one of the reasons why this is because iron oxides and aluminum oxides and clays, they have a really high reactive surface. So they have a high surface area. This high surface area and their functional groups that are present on clays can react with all the functional groups present in organic matter. So what that means is usually soils that have a higher amount of clays will have a higher amount of organic carbon and organic matter. So in order to kind of test that theory, one of the master students in our lab chose to do a project where she looked at many different soils that had many different soil orders. So we had inceptisols and mollisols, alphasols and altisols. So these are all site locations at the mesonet stations. And these soils were kind of, spanned a range of rainfall. They spanned a range of soil pH, as well as many variable depths. So we took those soils and started looking at all their aluminum and iron fractions, different mineral contents, just to see if there's any positive or negative correlations with organic carbon in the soil. And some of the stuff that we found, so if we just simply look at overall of the soils that had an acidic pH below a pH of 7, we get pretty decent trends with iron. So as your iron content in your soil increases, the amount of carbon the amount of carbon percent in your mineral associated organic matter linearly increases. So that's kind of pretty typical of what you would imagine, and other research has shown that. However, there were some interesting results seen with ions like magnesium. So in your basic soils, so our pH soils that were above 7, we actually started seeing a decrease in organic carbon when your magnesium in your soil increases. So some of that kind of brings up the question of why. We're still kind of working on that, but some of our I guess hypotheses are that when these clays, when your clay percentage in your soil that has a high amount of magnesium, when the clay percent in your soil is so high, it actually starts to limit your root growth. And so if your clays are really, your clay percent in the soil is really high, you start to see less, root mass and overall less biomass. So the amount of organic carbon in the soil is decreasing. So that's one of our theories on that. But we're still kind of working out to see if there's some, maybe there's some clay percentage that it actually increases, but then when it gets so high, your organic carbon decreases. So that's something we're still working on. So the last thing that I'll kind of talk about before priming is, so this project is kind of in its infancy, but we're looking at organic matter and organic nitrogen mineralization rates. So, what soil and environmental properties actually control the rate of nitrogen mineralization? So we're primarily focused for this study on organic nitrogen. So we're looking at different things. So one of the things is how does your particulate organic matter and your mineral associated organic matter nitrogen ratios. So if we alter those ratios, so if the soil has almost all the nitrogen, the palm fraction versus the mineral associated organic matter. So we're looking into those ratios to see if that actually influences the mineralization rate. Other things that we're also looking into are the abundance of minerals. So if the soil has more reactive 2-to-1 minerals versus 1-to-1 minerals like kaolinite, what does that mean for nitrogen mineralization rates? As well as other things that we're kind of looking into are microbes and microbial diversity. So what is that kind of role do microbes and the diversity of microbes play on mineralization rates? And the other thing that we're looking at some is water content. So how does water content, influence SOM in the soil? And this kind of has implications in the soil fertility and nutrient management sector. So so the more we know about nitrogen mineralization rates, hopefully that'll lead us to have less reliance on nitrogen fertilizer application. So if we know enough about this mineralization rate, then maybe we can hopefully predict how much nitrogen will come available for your crops during the season. So if we know how much will actually come available, then you may not have to apply as much nitrogen for your crops. And then along the same line of thought, so if you're not applying as much fertilizer, then hopefully you're saving money. And with that, the last thing I'll talk about to kind of set up Dr. Gilling and her presentation is the soil priming effect. So how many, by show of hands, have heard of soil priming? Nice. Good. You'll get a good presentation very soon. So soil priming. So I want to introduce this concept through a study done by UC Davis. There's a lot of studies that actually show this, but this one was a fairly recent one that had a really interesting result. So just a brief background information. So this is a long-term trial study that as of now it's around 20 years old. They're going for like a centennial farm type thing on the UC Davis campus. But right now it's 20 years, I think. but conducted on the UC Davis farm, and they wanted to investigate organic carbon changes in response to different cropping systems and organic matter input. So on this farm, they have many different types of cropping systems. They have wheat fallow systems, wheat fallow with winter cover crops or other cover crops. They have maize and tomato. They have maize, tomato, wheat. So they have a lot of different types of crop rotations paired up with winter cover crops, as well as different things like compost. And one of the cool cropping systems that had a really neat result was their maize tomato rotation with winter cover crops. So their cover crops consisted of legumes and oats, and what they saw over 20 years of cover cropping, so cover cropping was actually really interesting. So they sampled the soil from 0 to 200 centimeters, from like 0 to 30, and then 30 to 200 centimeters. So really, really deep soil samples just to look at the organic carbon concentrations. And what they actually saw, so after around 20 years, they had a 3.5% soil organic carbon increase in their top 30 centimeters, but a 10.8% decrease in their 30 to 200 centimeter depth, which led to an overall loss of soil organic carbon. So this is 20 years of consecutive cover cropping, leading to less organic carbon than they started with. So that brings up a couple questions actually, is why exactly did this happen? And then what depth should we sample when we need to monitor organic carbon concentrations in the soil? So the main question, so why did this happen, is the concept, so soil priming. Dr. Gilling will talk about that in our next presentation. But primarily their thought process on this is that all the cover crops over the years, when they break down, they release these organic acids that's leaching through the soil, and that's activating microbes that are at depth. So when these microbes at depth get these, pretty soluble nitrogen kind of rich compounds, it activates them, they get hungry, they consume those, but it gives them enough energy to not only consume that from the cover crops, but they can still continue to work and start to degrade native organic matter, hence your loss in organic carbon. So, so just something to kind of think about. And so the key points for today's presentation that I want to kind of drive home. So your organic matter functional group chemistry, it kind of mediates virtually all the properties of your organic matter in your soil. There's kind of a paradigm or a shift away from soil humidification theory, which is the old school method, and shifting towards a more, the new school method of POM and MAOM classification, as well as organic matter forms via all the inputs, so plant microbes, plant exudates, so all these kind of come together to form organic matter. And then the last thing, so in general, mineral-associated organic matter is considered more stable. However, there are some things like soil priming that can alter this kind of thought process. And with that, if anybody has any questions, I'd love to hear them.
One quick question.
Yeah. Why didn't they use the crop that was people good at that change that full thing?
It could have, yeah. So in this study, they only looked at, I think, wheats, and then legumes were hairy vetch and I think faba beans. But that's kind of what they do in California. But there were other treatments here where they also, to supply their nitrogen rates, they used composted manure. That actually showed actually good results where they actually increase organic carbon over time. So at all the way from 0 to 200 centimeters. So there's definitely things, I don't want to leave on the fact that if you cover crop and your organic carbon disappears. There's, that was just in that instance, but different cover crops would possibly lead to a different scenario, as well as different things like organic amendments.
Thank you, Andrew.
Yep.
All right, our next speaker is Dr. Euling. I'm going to let Andrea introduce herself. You do a better job than I can. Here's this. You're now on. Yeah, you can go anywhere you want. And I'm going to turn this off.
All right.
Okay.
How many, sir?
Huh?
How many minutes left?
7 minutes left.
What do you mean left?
Oh, till you're 45, 35 minutes in.
Okay.
38 minutes
So you'll
I'll
stand up when then. Okay, perfect. Thank you. All right, how's the sound? Can you hear me okay? Is that all right? All right. Louder? Okay, maybe I don't want to shout, but I don't know if the volume can go up a little. Okay, so let's pull this up. Let me test my slides. All right. Okay, hi everyone. It's really nice to see you all. Really great to be here. My name is Andrea Yilling. I'm an assistant professor of environmental soil chemistry here at Oklahoma State University in the Department of Plant and Soil Sciences. So today I'll be talking about this practice of adding liquid sugar to soils. So there's a lot of increasing interest in this. Gotten a few questions. I was on a podcast with colleagues about this, talking about sort of the uncertainty around this. A lot of products out there that make a lot of claims around, boosting nutrients, improving your productivity, and there's unfortunately not a lot of peer-reviewed research, very limited field trials about this. And so what I'm doing today is drawing on my own research that's mainly based in the lab, and some research from others that's mainly, again, based in the lab, a bit in the greenhouse. But from these research experiments, we can draw some insights, I think, that we could apply to the field to think about possible consequences, potential benefits, and hopefully by the end of this, you'll come away with a few new concepts and a new understanding of sort of rhizosphere processes. So I'll get into that terminology in a bit. But first, as I said, there's many liquid sugar products out there, more and more on the market with many purported benefits. So, they'll say they're going to increase your soil microbial activity. They're going to increase absorption of essential nutrients. You'll have instant energy to plants. It'll increase fertilizer uptake. It's relatively easy, cost-effective relative to other inputs, and environmentally responsible or benign at least. So I want to first make a distinction between foliar and soil applications of sugar, because all of these products will give you instructions for how to apply as a foliar application or in furrow, in your irrigation water, and so forth. And my interpretation is these are very different, the effects that they might have, whether you're doing a foliar application or a soil application. And also, the majority of the evidence from field trials is really on these foliar applications. So the research that is out of a few different extension offices are showing inconsistent effects on yield. So generally no effect on yield, or kind of spotty positive effects here and there depending on the crop and the system. But what you do see with these foliar applications, there is some research out there, as you see on the right here, that there are possible benefits to your insect ecology in your system. It might boost some beneficial insects. So on the right, what we're seeing is in an alfalfa system, we had some plots that were receiving sugar, some without. And those that received sugar in this study, they found that it increased the residence time of lady beetles by a few hours compared to those plots that did not receive sugar. So potentially just retaining beneficial insects for a little longer in this alfalfa system. So just an example, there's a few other studies like this out there. There's also studies showing that sugar additions above ground kind of trigger a plant defense mechanism as well. So there might be sort of protective response. I do not study the above ground plant response, so I'm not focusing on these foliar applications, but I wanted to highlight it because there are a number of studies out there, and there is a really good summary by University of Nebraska Extension on these above ground benefits and some of the uncertainty around it. So I just wanted to highlight it to make you all aware that there is some information about that out there. But I'm a soil scientist, and I'm also a lot more interested in talking to you today about these soil applications and their effects on possible soil health and possibly crop productivity. Unfortunately, like I said, there's limited peer-reviewed research on how sugar amendments might affect soil health and crop productivity at the field scale. But what I'd like to do today is make connections to a lot of lab-based research that's out there that is on specifically rhizosphere processes, the zone of soil that's surrounding roots, where you get naturally occurring sugars that enter soil below ground from roots. And so these sugars that are naturally being released into soil, there's a lot of research on this and its possible and its real effects on soil and plant health. And what it's causing is this priming effect, which Andrew defined for us and alluded to at the end of his talk. So this priming that sugars cause is a real phenomenon that's well studied in the lab and in the greenhouse primarily. And there are certain lessons that we can draw from this research on root-driven priming and its short and long-term effects on nutrient cycling. Okay? So how and why are sugars applied to soil? So first off, the recommendation is to apply sugar to soil as a pre-plant broadcast in furrow or in your irrigation. And in theory, the idea here is that by adding sugar to your soils, you're fueling microbial processes. It's going to release nutrients from organic matter. It's going to give your microbes a boost, maybe a bit of a head start, and make nutrients possibly more available before and or as plant nutrient demand increases. Okay? And what I find interesting when I think about this practice is that it really, in effect, is simulating a naturally occurring process, something that's occurring constantly in soils with all plant species that are alive and growing in soil. And this is this rhizosphere priming process that's driven by plant-derived sugars. So my goal for this talk is by the end, you will be able to define rhizosphere priming and have a general understanding of its overall importance, its effects on the soil environment, and with that information have some insight that you can apply to what's possibly going on in these soils when I apply sugar. What are possible potential benefits? What is the uncertainty? And what are possible consequences? Okay, so first let's just talk about sugars and where they are in these plant soil systems. Well, they're everywhere. They're abundant. Sugars are the most abundant organic compound in the biosphere. They're, of course, the basic building block of polysaccharides, so cellulose, hemicellulose, starch, pectin. These compounds make up the bulk of your plant tissue and your residues, especially cellulose. And the building block of cellulose is glucose, is a sugar. And so when you have residues sitting on your soil surface, and as they become broken down by microbes, they are eventually converted into their basic building blocks, these sugars. And so these are constantly entering soil as your litter is being decomposed in soil. So this is one major pathway by which sugars enter soil. The one I'm talking about today is the sugar entering soil from root tips, live, actively growing root tips. So all plants, all species, as they're growing in soil, they are releasing compounds into soil at their root tips. Okay? So they're not only taking up nutrients, they're also releasing and exuding compounds. Often, and most often, they're very simple, very small, simple carbon compounds that include sugar, amino acids, organic acids, very small compounds, right? And as you see on the right, all of these compounds collectively are referred to as root exudates, all right? And they include very small compounds, larger compounds like enzymes, and each species, plant species, will differ in the amount that they exude, what they exude, but it's in general a very complex, diverse mixture of compounds that they're releasing, okay? And then I also want to quickly also define the rhizosphere, which I've mentioned here. So the rhizosphere is specifically the area of soil under the influence of a root. We're talking, 1-2 millimeters away from a root, okay? This is the zone where exudates are being released. It's causing a lot of changes, localized changes immediately around the root, and this is the zone that is referred to as the rhizosphere, okay? And so what I think is a really interesting fact that is often overlooked, if you think about a plant growing, it's fixing CO2 from the atmosphere, using energy from the light to create its own plant carbon, right? The tissues that you see, the roots below ground, all of the carbon that goes into plant carbon. 20 to 40% of that plant carbon is actually being released below ground through exudation, okay? It's a massive amount of carbon. You don't see it, but it's really a massive amount of carbon that they are pumping. Plants are pumping carbon into the soil, okay? As simple, very simple carbon exudates, where over 50% of the exudates that are being released by roots are sugars, sugar compounds, really simple sugars. And these are feeding and fueling the microbes. They're priming the microbes. That's where that word kind of comes from, where, you know, as with us, if you were to eat a cookie or a simple candy, right, that's going to give you a boost of energy. Same concept with microbes. And so these root exudates will accelerate nutrient cycling at a really localized, concentrated scale, where in the rhizosphere what we often see are shifts in your overall soil solution composition, the nutrients, the ions that are there, the soil pH. You'll see shifts in your microbial community. So in your rhizosphere, you could see a very different population of microbes from what you see a few inches away from the root. You see an increase in enzymes being produced. So these microbes, when they receive these sugars and organic acids, they're going to ramp up their enzyme production. Okay? And all of this collectively can lead to increases in organic matter decomposition. Okay? So all of these sort of stimulatory effects in general that you see in the rhizosphere are sometimes referred to as the rhizosphere effect or the priming effect, or the rhizosphere priming effect. So that's where those terms come from, okay? And so, as I've mentioned, sugar exudates are really a simple source of energy for microbes. When they're fed a simple sugar, like glucose, most microbes will immediately ramp up their activity, okay? They're going to be respiring a lot more CO2. They're going to be producing a bunch more enzymes, just sort of accelerating their cellular metabolic activities. And so often what you'll see is an increase with this microbial activity. You see an increase in the enzymes that they're producing. These enzymes are then going to attack organic matter and release nutrients, all right? So that's what we often see in the rhizosphere, is this cascade of processes. And so what is the overall magnitude of the priming effect? It can be massive, where living roots can enhance decomposition rates up to 380% compared to decomposition in root-free soils. And what we're seeing on the right is, we're seeing on the right is sort of how priming really occurs in all systems, but at different sort of magnitudes, where we're looking at just the overall extent of priming. So this dashed line means that there's no priming effect in these systems, but anything to the right is a significant and positive priming effect. And we are seeing that differing systems, grassland crops for instance, they have different sort of overall magnitude of priming that is observed, okay? And you also see differences in your between soil textures. A finely textured clay soil in this study that looked across 31 experiments that were conducted, they synthesized across all these studies and found that the soils that were more finely textured had overall higher rates of priming observed compared to more coarsely textured sandy soils, okay? So there's some interesting differences by soil type. It's not always driven by the plant necessarily, all right? And another sort of fun fact here I want to highlight is that if you think of just a soil and all the CO2 that's being released from it, a massive proportion can actually be derived from this root-driven priming. So in this study, it was a temperate forest soil, but what they found that out of all of the CO2 that's being released, up to, or about a third of that CO2 is attributed to priming. So you might think, at the root tips or actively growing root tips where the rhizosphere is, it's really quantitatively not a very large volume of soil that it occupies, right? Compared to just your overall soil profile. But it actually has this really disproportionate effect on your carbon, respiration of CO2, on the nutrients that are released as ammonium and nitrate and so forth. So it really, for how small of an area it occupies in soil, it has this really large effect, priming. And so rhizosphere priming is known to enhance multiple microbial properties and processes. So we're looking at a similar similar graph to what I showed before, where the 0 line is showing no effect in the rhizosphere relative to the bulk soil. So non-rhizosphere soil is bulk soil. So anything to the right of this 0 line is showing a significant difference, an increase in a certain property in the rhizosphere relative to non-rhizosphere soil. And we see all of these properties are in this positive sort of rhizosphere priming zone over here. We're looking at different systems, but I just want to point out we have microbial biomass that just represents the overall size of your microbes, microbial respiration, so the release of carbon as CO2, the enzyme activities, nitrogen mineralization, organic matter decomposition. So all of these overall effects of priming on microbial processes are generalizable. You see it across different systems, and you see it with respect to many different measurable soil microbial properties. And I will just note that a lot of these properties are part of sort of the suite of soil health metrics, okay? So you could also think of priming really supporting soil health in a way, okay? So I want to show some work from my own research here where, oh, and I'm sorry about the labels down here. You don't have to worry about them. Just know that these are different types of enzymes, that these are enzymes that release carbon, say release glucose from cellulose. These are enzymes that release nitrogen from nitrogen-containing compounds. These are oxidative enzymes that are more general enzymes that can attack many different types of organic compounds. But what I found here is when I simulated priming, I added glucose to soils. I then measured how much the enzyme activities responded, how they were changing in response. So we have in gray is the soils that received just water, it's a control, and in orange we have the soils that received glucose. So I'll just focus on the gray and the orange for now, don't worry about the green bar. But what we see is this, in many cases, this big increase in the enzyme activities in both carbon acquiring and especially nitrogen acquiring enzymes relative to the control. So there's this, clear response in enzymes, important enzymes that are responsible for releasing nutrients from organic matter, okay? And I will say that a lot of the research that I'm drawing from is really these simulations where we're trying to recreate the rhizosphere environment in the lab. So we take soil, put it in a mason jar, and we drip, drip, drip a little bit of glucose solution on it to simulate what would happen in the soil, okay? So very controlled, in many ways artificial, has its own drawbacks for sure, but we can learn a lot from it. So some of the other slides coming up are similar experiments to that, okay? These simulated exudates, simulated rhizosphere. But what I want to highlight here is that sure, you have an increase in these microbial processes, an increase in respiration, mineralization, and so forth, but what does that mean? What are the consequences? So what you can often get is that you'll, over time, due to priming, get a net loss in carbon or nitrogen. So what will happen is that, say you have on the left here a soil that has no glucose, no roots, no nothing, okay? It has just this baseline amount of CO2 that it's releasing, okay? That's the yellow here, okay? The carbon, say that's this yellow here we'll think of as the CO2 that's just coming from soil organic matter, okay? You take this soil and you add to it glucose, all right? A certain amount now of the CO2 coming out of the soil is going to be coming from that glucose, all right? So this green bar here is showing the glucose-derived CO2. But then you're going to have this boost in activity by the microbes. So these microbes are actually going to not only decompose and respire, release the carbon from the glucose, they're going to be ramping up their decomposition of the soil carbon. So if you look at this difference in the bars here, this area, this little rectangle is in here, is showing the extra carbon that's released as CO2 due to that addition of glucose. So you can imagine that over time, if you have this continue, you'll have actual losses of carbon in your soil, all right? So it's basically, think of it as the microbes mining for nutrients from organic matter. Yes, they're decomposing the glucose, respiring it, but then they need more nutrients for their own needs, nutritional needs, so they're going to go and mine nutrients from the organic matter, leading to losses of nutrients from organic matter, okay? So the extent of priming, something that I think is especially relevant to these field applications of sugar is that the extent of priming that you'll see can often depend on soil fertility. So if you have a soil with low fertility, what some folks are finding is actually they'll have higher rates of soil organic matter priming relative to a soil with high fertility. So this is just a gradient going from low to high fertility soils, and we're looking on the y-axis of just think of it as the magnitude of priming. The higher the bar, the more priming that exists, okay? And don't worry about, for now, the single and repeated, the red and blue bars, don't worry about it. Just the overall curve here, notice how you have higher rates of priming in low fertility soils. So if a microbe is receiving sugars, which has no nitrogen, right? They're going to receive sugars. They need nitrogen. So they're very active. They're increasing their enzyme production. They're respiring CO2, but they need nitrogen to keep going and to continue thriving. So they seek nitrogen from the organic matter. So in a low fertility soil, they have to do a lot more digging and mining to get nitrogen to meet their own nutritional demands, if that makes sense. So a lot of this comes down to the microbes and their nutritional needs. But also, I think is important to think of, if you're adding sugar to low fertility soils, this could actually cause really extensive, well, over time, could lead to more extensive losses of nutrients in your soil. So sure, you're releasing nutrients, potentially boosting microbial activity, but I think it's important to think of, what the net effect is on your fertility and how it interacts with your existing fertility. So the frequency of your glucose additions will also affect your net carbon balance, whether you have overall no effect on your total soil carbon or if you get a loss over time. Now this may not be super analogous to what's going on with field applications of sugar, because I think many people are just adding once or twice in a season, but who knows, there could be people who get really excited by this and want to add it multiple times throughout a season, and that can be problematic, I think. And what we're seeing here is a difference. We're comparing occasional and frequent glucose additions. This is just comparing frequency. We just add it a couple times versus if we add it every month, okay? And what we're seeing is the net carbon balance. So if anything is below the 0 line, we're losing carbon. We're losing soil organic carbon. And what we see here is that with more frequent inputs of glucose, we get this loss of carbon over time, all right? And similarly, we see that in the study over here where the repeated versus single addition. So in blue, we have multiple additions of glucose. In red, it's just a single addition of glucose. Again, looking across that fertility gradient. But below the 0 line, we're seeing a loss of carbon. So the low fertility soils with repeated additions of glucose, we're seeing losses of carbon. Okay, so again, this interaction with your soil fertility, the importance of how frequently you're adding sugar to soil can really influence your overall fertility or overall nutrient storage capacity in your soil, all right? I also want to talk a bit about how rhizosphere priming can encourage microbial mining of nitrogen from organic matter, which can be good in a way that soil organic matter contains so much nitrogen, right? But it's locked up. It's not immediately available to plants, right? It needs to be released, decomposed, and converted into ammonium and nitrate. So if microbes are mining nitrogen, then they could, in theory, release that nitrogen and make it available, more of it available to a plant. But what many are finding is that it's not certain if this nitrogen will be made available to the plant at the right time or the right place necessarily. A lot of that nitrogen may remain locked up in the microbes, so it may not become released. And a lot of this comes down to plants and microbes effectively competing for nitrogen. They both need that inorganic end, that ammonium and nitrate. So there's more research in our group. We're looking into this as well. But I would say that if you're especially adding sugar to soil before the plants are actively taking up nitrogen, I'm guessing that a lot of the nitrogen that might be released would never make it to the plant root. But again, that's just my hypothesis. I don't have data to support that yet. Okay, and I want to make some connections also to the organic matter fractions and pools that Dr. Whitaker told us about. So just a reminder here, remember there's your total soil organic matter is complex and heterogeneous. There's a whole diversity of compounds that make up what is soil organic matter. And so in our lab, we're often separating organic matter into these two functionally distinct pools that are referred to as particulate organic matter and mineral-associated organic matter. And these kind of lie on a gradient of decomposition. So plant residues that are first entering your soil as stem, roots, leaves, and so forth, they start becoming broken down. So when you pick up soil and you see those leaves that are somewhat decomposed, right? Maybe the veins are remaining, or you see some bits of roots and whatnot, that is what makes up particulate organic matter, all right? That material, as it becomes more and more decomposed, smaller and smaller in size, it becomes associated with silt and clay particles and becomes part of this pool known as mineral-associated organic matter. And so this more decomposed stuff that's in mineral-associated organic matter is generally thought of as slower, more passive. Once nutrients enter this pool, it is locked in, okay? This is generally not accessible to plants. It's a really important reservoir, long-term reservoir for nutrients in your soils. And it's protected, and largely protected from degradation. And this is in contrast to particulate organic matter that is this more fast cycling and more readily available source of nutrients for your plants. And so what the relevance to rhizosphere priming is that what many are finding and what we're finding in our group is that priming may disproportionately affect this more slow cycling, stable, mineral-associated stuff, okay? And this has consequences too if you think about it, where if you were to repeatedly add sugar to soils, and if you were to potentially lose carbon from your soil over time, you might be affecting this really critical long-term reservoir for nutrients, okay? So just something to consider here. And I wanted to show some evidence to support that. So here we're showing how, or another group is showing how the frequency of the glucose input can affect the form of soil carbon that's decomposed, where if we look on the right, we're looking at frequent inputs of sugar versus just a couple inputs, occasional inputs of sugar. And we're looking at the cumulative priming, just the overall priming effect over time in this incubation. And they're comparing decadal cycling carbon versus fast cycling carbon. So we can make this analogous to mineral-associated, the slow, more passive, stable, protected carbon versus the fast cycling particulate organic matter material, okay? And what we're seeing is that with frequent inputs, there's much, much higher priming rates in this stable, relatively, otherwise slow cycling pool, okay? I wanted to also briefly, how are we doing on time, Dr. Arnall? Am I okay? Okay. All right. I don't know, maybe 10 minutes, we'll see. Okay, all right, thank you. So I just also wanted to highlight some other research, a little bit of a shift, but it still relates to priming effects on mineral-associated organic matter, but it's in the context of a changing climate. So in many parts of the US, we're already seeing it. There's shifts in sort of the precipitation patterns. You'll see intense rainfall, longer periods of drought, just these wet-dry cycles that are certainly a shift from historical patterns in precipitation. And so these wet-dry cycles in soil, we're trying to understand how it will affect your overall soil's capacity to store and sequester carbon. But what I'm really interested in is how this interacts with the priming effect. So will carbon that's formed under this shifting climate, could it potentially be more vulnerable to glucose inputs, to priming and these processes we've been talking about? And so what I've done in a number of studies is basically separate out the organic matter that's the finest mineral-associated silt and clay size stuff. So that's the mayom here. And this is just showing the sort of sequence we use with dispersing soils, putting it over sieves to really separate and isolate the silt and clay associated organic matter. We mix it with sterile carbon-free sand, put it in a jar, and measure some stuff and simulate rhizosphere priming, okay? So relatively, very artificial, that is a fact, but there are some sort of basic mechanisms we can learn from this. But what I want to highlight is in this study, we took these mineral-associated organic matter sand mixtures, and we simulated wet-dry cycles. So these fluctuations, say in precipitation you might see out in the field, we tried to recreate that in the lab. So we took some of these soils and we put it through these wet-dry cycles over 3 weeks. Some of these soils we kept at constant moisture, all right? And to these soils we added an amino acid that had a label, a tag. Think of it as, a tag in the carbon and nitrogen that allows you to track and trace specifically the carbon and nitrogen that's in that amino acid in the soil. So say you have 10 units of amino acid. Here, you could say at the end of that experiment exactly how many units are left of that amino acid in the soil. So it's a very cool tool. We use a lot of stable isotopes in my lab. But what we were interested in is if you were to add this very common compound that enters soil, you add it to this soil and then impose this moisture treatment, we want to know sort of the overall fate of that organic matter. And look at how at the end of this incubation, is the organic matter that's formed with this amino acid, is it potentially more vulnerable to priming? So just to visualize it, we have this original soil, we add this tagged amino acid, we put it through this wet-dry cycling period so that at the end you have now a mixture, the original sort of older carbon, and now you have some newer carbon that's derived from this amino acid that we added. So the question in this study was, is the mineral-associated organic matter formed under wet-dry cycles potentially more vulnerable to priming by exudates? All right, and so I will skip this, but I will just show the sort of take-home message in these slides here is that what we're looking at is the CO2 that was respired in these soils, and we're looking at, on the bottom, the different priming treatments, which for now just compare the control, which is just water, against the GLU is glucose. And the two different colors are showing in brown, that's the wet-dry treatment, and the bar below it, the blue, is the constant moisture. And on the left, we're looking at the CO2 respired specifically from the amino acid that we added. Remember that tagged compound? We're seeing how much was respired of this newly formed organic matter. And we see that the newly formed organic matter that formed under wet-dry cycles is much, much more vulnerable. It's being decomposed at much higher rates than organic matter formed under constant moisture. And we see actually the same thing occurring in the older pre-existing organic matter, where the difference is not as striking, but what we see is that wet-dry cycles, these sort of water stress treatments in a way, are really ramping up the priming of these otherwise stable forms of carbon, okay? And so what sort of I'm taking from this is that newly formed organic matter may be more vulnerable to priming and have less opportunity to enter these more stable, persistent forms of organic matter. So I, would think, if certain soils are repeatedly over time exposed to wet-dry cycles and extreme moisture events, could they be over time losing organic matter from these mineral associations? Okay, so let's back up. What can we learn from these many lines of evidence from lab-based research on sugar effects on soil? Well, yes, adding sugar to your soil likely is going to increase microbial activity. That is a fact, probably generalizable across your soils and systems. But it's uncertain whether this will translate to increased nutrient availability at the time and place that the plant needs it, okay? And at the rates supplied by these sugar-containing products, the sugar is likely used by microbes within hours to days, sometimes minutes. So you're really adding trace amounts, and this stuff will disappear, okay? And many products also, I've noticed, contain other biostimulants and nutrients that, in my opinion, probably have a larger effect than the sugar in that product. And I will stress, limited peer-reviewed research on this, so I just want to underline that. And then with repeated use, there's also maybe the potential for loss of total soil nutrients over time, but this of course depends on the rate and frequency of use and the fertility of your soil, all right? I also just want to highlight that what's going on in the rhizosphere with priming is at a spatial scale so different from what's occurring when you're broadcasting sugar across a field. So there's some important differences there. But we need more research on how we can promote the rhizosphere effect. Plants are releasing sugars naturally at the time and place where they're actively taking up nutrients. So we need to be able to build inherent fertility in soil and optimize fertilizer management and really promote conditions that favor these natural root-induced priming processes that potentially release soil nutrients. With that, I will thank you for your time, and my email's there, so feel free to reach out if you want to follow up with questions or thoughts, anything I can clarify, but I wonder if there's any time for questions? Okay, awesome, good.
Any questions for Dr. Yelling? Mike, comes right up. Mike's ready.
Oh, over here.
Basically using the sugar as a carbon source to feed the microbes.
Exactly.
Would you get the same response if you use, say, used oil, fat oil, waste fat oil?
Sure.
As a carbon source?
Yeah, you'll get a
Are you going to affect your carbon degradation differently?
Yes, and likely yes. I mean, it's a carbon source. Microbes can use that. They will respond to it and probably increase their activity. What will differ is sort of the timing of the response and how large that response is, because they probably can't use it as immediately as, say, sugar. They can right away turn that into catabolism and all the metabolic processes. But what you're describing, that sort of input, I would imagine there'd be a lag, and there'd be other processes at play. So there's a lot of research out there where they do just that, is compare different types of carbon inputs and see how the response differs. So wish I had another 30 minutes, but yeah.
And when you're talking adding sugar, are you talking cane sugar or?
All different kinds, like glucose, sucrose, dextrose, beet sugar derived stuff. So yeah, it's a whole range of sugar compounds. Yeah. Yeah.
First, I want to thank you for the most intriguing presentation in 5 years of this winter crop.
Wow. Is that
I have a lot
for
of
real?
questions, but the one question I want to ask you is we've got this thing facing our industry on carbon sequestration. And it really threw me for a loop when you talked about the loss of carbon in the soil. Surely you're not taking into account the residue contributions back to the soil.
So in many cases they are accounting. They're doing a full budget of the carbon, and they can show that simply having live roots can lead to a loss of carbon. So there's some interesting studies where they have these detrital inclusion removal treatment plots. The acronym is DIRT, but there are these replicated experiments around the world where they kind of include or remove residues, include live roots or don't include live roots to try to account for the effect of live roots. And they show that having live roots can actually lead to losses of carbon despite inputs otherwise from residues. But it's not across the board. You'll have cases where your residue input can offset the potential losses from the rhizosphere. So I have really been hyper-focused in sort of isolating this rhizosphere priming effect. So I don't want to alarm people that it's leading to these massive losses of carbon. Sometimes it's really subtle or no effect. So yeah.
All right, I think for time's sake, quick question. Is soy soap considered sugar?
I'm sorry?
Soy soap considered sugar?
Soy soap? I don't think so. I don't know. I'm not familiar with that product. Is that? I'll look it up. I'll see if I can look it up and follow up with you. So yeah. Thank you.
All right, thank you, Ivy.
Yeah, thank you.
Go ahead and hand it to Jordan.
Okay, sure.
So yeah, so I want to brag on Dr. Yealing. She's relatively new. She's not that new, but we have to have her here. I think she's one of the best out there in the nation, the world, as far as what we got here. So I'm excited to have her on the team in our thing. So, but with that, we're going to bring in another top shelf. This is actually, Dr. Yealing said, we got to get Jordan here. So Dr. Wade, no, that's true. Dr. Wade out of the University of Missouri, soil health. I had your thing pulled up, but I've dropped it, Jordan, so you're going to give yourself a little bit of a...
That's all right.
My phone was late. So with that, continuing on with the last section, and Jordan, I'll kind of stand up. We got about 5 to 10 minutes left, okay? But you're free time. You're our guest.
Sounds good.
All right. Clicker's right there, and point forward.
Perfect. All right. Cool. And we're good. So my name's Jordan Wade. I'm a professor at University of Missouri. I do soil health, soil fertility work there, and I'm also director. We have a soil health assessment center. So similar to a lot of universities have like a soil testing lab, soil fertility lab, we have one that focuses primarily on soil health, and I run that. We do basic soil fertility stuff, but then we also have this kind of additional layer of things that we look at. So I run that center as well. But what I want to talk about is, you know, everyone's talking about soil health these days, and people have lots of ways that they like to measure it, and not all of those it's not always clear how to use that in your day-to-day decision making. So that's kind of what I'm here to talk about today, and also kind of give you a heading of where we're going, and yeah, hopefully that'll give you some advance notice when we start to think about this and have some better results in a couple years. So, where do I got to point this thing? There we go. All right. So first off, why should you assess soil health in the first place? This is probably the most basic question that we can start with here. And the reason is because you're out there, you're doing some sort of management on your fields, right? You're trying cover crops, you're trying reduce tillage, you're trying different rotations, etc. And you're doing that because you want something to happen. You say, I want to change my rotation because I want to reduce disease pressure, right? I want to reduce pest pressure. I want to increase organic matter. I want the end bump from the legume, something like that, right? But the thing is that management, if that's going to actually give you that outcome, it varies from place to place and from field to field, right? Can vary by a lot of different things. Vary based on climate, can vary based on geology. Doctors Whitaker and Yilling were just talking about that soil mineralogy and how that comes into play. But it can also matter how long you've been doing that management, right? And it can also based on things like your soil texture. So all of those things, that's a lot of things to think about all at once for you, right? And you want to know if management that someone tried across the state works for you. So to kind of bridge that gap and serve as decision-making tools, we have soil health assessment. Now that is comprised mainly of soil health indicators, which you may or may not be familiar with, but I'm going to talk about a couple of them here today. So that assessment helps us kind of bridge the gap from the context specificity to give you, tell you, is the management that I'm doing heading me in the right direction, getting me the outcome that I want, right? My job as a scientist is to make sure that those indicators are reliable and that they're going to work for you. So first thing I want you to know is that assessment piece, that's a tricky business, okay? We've been working on this for a little while, and soils is hard, and it varies from place to place, and so this is why when you're filling out, like for NRCS, your soil health management plan or something like that, the SEMA 216, this is why they give you a ready-made set of indicators that they want you to measure, right? They say, we want you to measure these things because these are the things that we've already worked on. So here we've got soil carbon, which is just total soil carbon on combustion. They've got an aggregate stability measure, this new measurement called ACE soil protein, which is your organic nitrogen in your soil. And then there's permanganate oxidizable carbon, or active carbon, some people call it. And then there's respiration upon rewetting, which some of you may be familiar with. It's part of the Haney test. So, but then the other important component is people. They want a qualified individual to help you interpret it, because again, this is a tricky business to do. Okay, so it's things like CCAs, certified professional agronomists, professional soil scientists, etc. Okay, and that's because we're not quite there yet on how to interpret some of these things and how to make sense of them. I want to focus first off on a couple of these here that I've done a little bit of work with. So the first one is this permanganate oxidizable carbon and respiration. So this is kind of soil health assessment 101. We use these specific measurements and we interpret them on a one-to-one relationship. This measurement means this measurement means this, right? And so this is kind of taking these measurements that we're using often and making them make sense. So like I said, this permanganate oxidizable carbon, so this is this purple here, And as that purple kind of disappears, that relates to the amount of carbon, the oxidizable carbon that you have in there. Okay? The other one is this mineralizable carbon, or respiration, or flush of CO2 upon rewetting. We've got all kinds of names for it. But the idea here is that this is the CO2 that comes off your soil after you rewet it. It's a component of the Haney test, probably the most prominent component of that. And then it also is, there's another company called Solvita that uses it. It's not an endorsement, just an example. A lot of people have heard about it, so I want to make sure that I acknowledge that. So when we take that, we want to say, can we use these to actually make a management decision, right? When you're doing something, you're trying out something new, it might take a couple years to see an effect, and you want to, as Rick Haney says, he says, you know, you need to have these measurements to give you the courage to keep doing that. You know, if it takes 3, 5, 10 years to see the effect you want to know at year 2 or year 3 if you're headed in the right direction. So this was some work that I did before where we took a bunch of sites across 10 different states. We had a little over 1,000 observations, and we said we're just going to throw everything at it. We're going to throw all kinds of different managements, right? All of these soil health building managements. So it's everything from reduced tillage to perennial systems, organic management with high organic matter inputs, composts, something that's kind of a blend between manures, compost, etc., cover crops, winter fallow, and variations in that rotation. So we really threw the book at it and we said, does anything fall out here? Well, what we found was that in 100% of our sites, that there's this one relationship that held. So I'm going to spare you a little bit of the details here, but I'll suffice to say that what we found was when you put these two things in a graph against each other, that when you have more of this permanganate oxidizable carbon, that those are the managements that are going to be building your organic matter, right? When you have this respiration, this mineralizable carbon, those are the ones that are building your soil fertility, right? We ask a lot of things of organic matter, as Dr. Gilling was just talking about. It can go a lot of different directions. On one hand, we want to build our organic matter. That gives us aggregation, that gives us water holding capacity, all that good stuff. On the other hand, we also want it to give us fertility, right? And those two things are mutually exclusive, right? You either use it or you lose it. You can't do both. And so this helps you know, is this heading me towards which of these am I heading towards with my management? Right? So if you have more of this permanganate oxidizable carbon relative to your mineralizable carbon, you're building it. You're stabilizing that organic matter, doing things like aggregation, water holding capacity, et cetera, et cetera. But if you have higher respiration, that's mineralization. That's your fertility, your nitrogen availability, things like that, right? And so personally, I was a little shocked to find this because hardly ever in science does something happen on 100% of your fields. And we found this across all of them. And we had a little bit of a gut check in the middle of it because one of the fields wasn't working the way that it was like, why is this one different? Turns out we just had to fully separate, you know, the effect of cover crops and no-till, and then it fell right into place. And we saw this same relationship. So it was a little bit of a gut check mid-project that it was in fact happening. So what we said here is that we can actually use these two measurements straight up, one-to-one. Permanganate oxidizable carbon probably represents something that's building organic matter, and your respiration, you're oxidizing, and you're doing, mining your soil fertility. Now, both of those could be good in the right context, and so we kind of decided not to say that one is a healthier soil and one is less healthy, but we just wanted to say these are potential outcomes that you might be looking for your soil health. The good news is that this is something that you can implement pretty easy, right? These are very sensitive measurements. We see changes in them in 1 to 2 years. And so what you could do is you go out, try it on that one section of field that's giving you a problem, right? Take the soil samples from there and from the place where you haven't done anything new, right? And compare the two. And that's how you get this direct comparison. And again, gives you the courage to know if you're headed in the right direction, right? All right. So then some other folks, we published this paper, and some other folks said, is that so, right? So they wanted to test this out on farm. So this was up in Wisconsin and Minnesota, and they published this paper, and they said, we're going to do this on farm, and we're going to try this out, okay? And so they again had all of these paired managements on each of these farms, tillage, manure, drainage, different rotations, etc. And they said, okay, how do these things relate to yield, right? If mineralizable carbon, that respiration, is fertility, that should be more related to yield, right? And if permanganate oxidizable carbon is not, if it's more other things, building organic matter or whatever, then it shouldn't relate to yield, right? Well, turns out they found pretty much the same thing. Now, this is not the way I would visualize it, but this is their graph, and they ran the statistics on it, and they found that, in fact, this mineralizable carbon has a stronger and more predictable relationship. This looks messy. Their statistical models accounted for differences in sites, etc. But suffice to say that they found a similar thing there. And so said, all right, cool, we're headed on the right track. All right, now this is all good because, you know, it tells us across lots of sites, right? But the real question is, can you change something within your site? Can you change your management and produce differences, right? We want to know about what you can do within a site to address problem spots in your field, or, you know, to do zone management, things like that. So this is some work. This is someone else's work. This isn't me. This is some work that Rick Haney did down in Temple, Texas, on the ARS station there. So what they did is they went out and they did a really intensive grid sampling, 35-meter spacing, so that's about 100 feet in between each grid sample. And they were going out, they were giving this Haney soil test score and looking at yield, okay? They were looking at corn yield. So what they found is that relationship is a little variable. So just to kind of walk you through here, this is the site, this is their yield map here, green being more yield, red being less, and this is their soil health score that they assigned it, all right? Now, first thing is that you see that soil health score tracks with their soil map here, right? So this is one soil type here, it's all mostly red. Here's another soil type, that's green, and this one as well, right? Now, that tracks pretty well with their yield here, right, visually. Well, let's just not visualize it, let's like actually put it together in a graph. And what they saw is this pretty positive relationship, right? Higher soil health score means greater yield, right? Not a great relationship if you look at that R squared, but it's at least something, okay? But when they parsed it out by soil type, right? They had 4 different soil types on this field. They found that those relationships aren't as consistent as one might hope, right? So here we have a positive relationship. Here it's pretty much a flat line. Here's slightly positive, and here it's even negative, right? So the way that we put that soil health score together probably matters, and it matters according to the soil type, right? And so this is just the word of caution. We got to think about how to evaluate soil health in a better, fairer way, right? Because the real question that we want to know is how much control do we have to take our unhealthy ground and change it to healthy ground, right? That's the real question here. Some ground's better than others, kind of inherently, and that's all good. But what we want to know is we want to know how to improve your soil health on your ground, right? That is the only relevant question. Now, this graph right here says that maybe it's just out of our control, right? This ground is better than this one, and the relationship is all kinds of messy. So this is the kind of thing that I look to address in my research program, and I want to walk you through one of the projects that I did, a couple of the projects that I'm doing on exactly how to tease this out, right? So there's this, it seems like a very basic question, right? But is a healthy soil more productive? Meaning, can you go out to your field and improve soil health and make your ground more productive? So this was some work that I did during my PhD. This is all mostly Eastern Corn Belt mostly rain-fed, but we did this similar approach, but we layered it on with some N-rate trials, okay? So we want to know, can you change your fertility, right? Can you change your fertility program if you're investing in soil health? So again, we use all different kinds of practices. We threw it at our soil health measurements. And so we had cover crops, we had leguminous cover crops. Some people were using radishes, some people using rye, using 3-way mixes, 7-way mixes, all kinds of stuff, okay? Then we all had differences in tillage intensity, everything from 60 years of no-till to really intense chisel plow, moldboard. We have differences in rotation length, 4, 5, 6-year rotations. And then we also had different manuring histories, right? We know that manure can influence soil health as well. So we had 30 sites across these states, and we had a lot of these were on farm, as well as a couple were on research stations, right? And so in terms of the drainage, the soil type, et cetera, we got a pretty representative sample. And they said these sites represent around half of the Corn Belt. So we got a pretty good bang for our buck here. But we ran into the same problem that Rick Haney ran into on his fields, right? Every field is different because that ground is a little bit different. We have different soil types, etc. Right now, people can deal with this in different ways. Some people, they just throw it into a graph, right? So, oh, sorry, let me back up here. So if these are each different fields and we've got a couple observations from each field, right? We want to make sure that we're comparing within a field fairly. Now, researchers deal with this differently. Some people throw it all in a graph, and they'll say, hey, look, more soil health equals more productivity. And that's fine, but if you look a little bit closer, right, at something like this, we see that there's this clustering by color, right? Because again, some ground is kind of better than others, right? So this is one thing, and you can see here that it's not super meaningful if we're looking at a single site, which is the kind of level that you're making decisions on. But another strategy that people use is they just say, well, I'm going to average all these observations and I'm going to put them together, okay? Now, neither of these is really perfect. This way, we are on this side, if we kind of just throw them all together, we're conflating inherent kind of site fertility with the effect of your management, right? And so we're kind of double counting and we're overestimating the effect of soil health. On the other side, if we're just averaging, we're losing that. Then we are losing the variation within the site, which is kind of the thing that's of interest. So we're underestimating the effect. So what we did is we kind of split the difference. And we said, okay, you've got all these different fields, right? And what we want to do is we want to let each of them start at their own individual place, right? Because again, good ground is good ground, bad ground is bad ground. And we want to know, how can you move the needle on your productivity? And so what we did is we said, we're going to let it start where it starts, and we're going to see how consistent that change is. As we improve soil health, what's our change in productivity? Now, this gets us to our second obstacle, right? Is how do we measure soil health? Now, everyone has different opinions on how we do this, right? But some things don't change, really. One is that we can't just go out and measure a single thing to assess soil health. We got to measure a couple different things, right? In the same way that like when we go to the doctor, they don't go and take your blood pressure and say, you're healthy, and you're like, I'm here for a broken leg, right? It doesn't work that way. Similarly, when we measure soil health, we want to go and we want to measure multiple things to make sure that the soil is healthy, okay? And then we got to figure out how to combine those. Most of the ways that folks are doing it right now is they just kind of say, well, we measured the things and these things mean soil health, and then we figure out how to combine them. We do some math and we average them, something like that, okay? And we get a score. Now the problem with that is that it's kind of arbitrary. What happens if I measure one different thing than you, right? Well, maybe you get a different score, and that's a problem, right? Especially because if you're going out and you send it to a different lab, that might cause problems, okay? Or maybe someone has a slightly different method of measuring something. But so we needed to figure out how to kind of square that circle, right? So what we did is we said, well, I think that when you do better management, there's some change in soil health right out there. It's out there in the world. And we don't know which measurements accurately capture that, right? And so we kind of flipped the arrows here. We said, one is there's soil health. We have some change in soil health. Which things should we measure to capture that? Okay, we didn't already decide which things we needed to measure and then figure out how to combine them. So that was the first thing. Now, from a mathematical perspective, this is similar to like if you're measuring intelligence, okay? So like how smart you are doesn't depend on your math score, doesn't depend on your English score, doesn't depend on how quick you can put a Lego set together, right? But kind of all of those things are a reflection of your general intelligence, right? And so if we were to only decide that, hey, only your math score and your English score matter, well, what about, you could say, well, but there's other kinds of intelligence too, right? So we want to flip the script and say that soil health is something inherent and we got to figure out how to measure it. So I'll spare you on how we kind of run through that process, but because it's a kind of quantitatively complicated process, but I'll just say that we kind of think about soil health slightly differently, okay? Then we don't give a single score, but we want to see how it changes relatively within your field. Remember, your field starts at the place it starts, and we want to see how you can move the needle on that, okay? So we're kind of bringing both those things together. This is called a latent variable analysis. But what we wanted to do then is bring it all under the same umbrella and be able to take all of these moving parts at the same time. So first, we bring it into this kind of, it's called a multi-level model, but the idea is that some things happen at the field level. Your precipitation generally happens at the field level, okay? Your temperature, your mean annual temperature, happens at the field level. Your clay content, broadly, especially if you're doing plots this size, generally pretty consistent within a field, okay? So those things, we want to make sure that we're not double counting those, okay? Again, we want to make sure that we're focusing on how does your management change soil health. So we want to say clay content, gone. Temperature, precipitation, those are kind of to the side. So that's what this level is. What we really, really want to know is how does your change in soil health change your yield, and how does that interact with fertilization rate, okay? So the important part is that this differentiates between your kind of inherent properties of your soil, your good versus bad ground, and focuses on how much you can change it. From a scientific perspective, this is really important because we are now teasing apart correlation and causation, right? There's all kinds of sayings around correlation and causation, but the important part from a scientific perspective is how much can we accurately, confidently, causally attribute to changes in soil health, okay? So all of this is a long-winded way of justifying what we did, and I'm sure you just want to see the results. So is a healthy soil more productive? And the short answer is yes, okay? The bigger answer is that when you change your soil health, it's positively influencing your yield. And the kind of ballpark that we arrived at was that if you get a 20% relative change in soil health, that your productivity, your corn yield here, should increase about 5%. Okay? And this is after accounting for differences in nitrogen rate and precipitation, all that kind of stuff. Okay? The other important part here is that fertilization increases soil health. Fertilization increases soil health. We can't just eliminate fertilizer entirely, nor would we want to, but some folks seem to think that, soil health can do all the work. Nope, we need fertilization to maintain soil health. And it can happen a couple different ways, through the residues that we were talking about before, right? But then also nitrogen has, especially nitrogen fertilizer, has a way of slowing down decomposition. So all of this together means that, yeah, we can invest in soil, health. And so we can kind of go two different ways with this. One is you can either decrease your N inputs and maintain yield, right? Or you can increase your max yield, keep that N rate the same, invest a little bit in soil health, and you'll get that yield bump, right? So that is what the takeaway is from this study. Now, this is all Eastern Corn Belt. It's corn, soybean predominantly. And so I moved to Missouri, and they said, yeah, that's nice, but what about here, right? Like, well, so according to what we did before, you know, we're trying to evaluate it fairly. It should hold. But they say, no, I want to see it here in Missouri, right? So this is some work that we're starting to do on soil health and nitrogen management. So we've got a couple different projects. Hopefully you guys don't think this is too irrelevant to you because I'm just right next door. But first one is looking at cover crops and nitrogen rates, right? Those two things interact. You know, you might need a little bit more nitrogen if you're using cover crops. You might need the right time, right? So this is a project that we're working on from our fertilizer control board, and we've got probably 20 nitrogen rate trials that are going on around the state. And in Missouri, we've got corn and soybean, of course, but then we've also got a couple other crops that we're bringing into the fold here. So we've got, of course, corn and soybean, but then we've got rice down in the Delta. We've also got cotton, and we've got peanuts down there. And so we're bringing those kind of into the fold here. And then we also have, recently the USDA did these climate smart projects. There's about $3 billion worth of projects, and this includes about 50% of each project goes to incentives for implementing these managements. So I'm part of one of these projects, and what I'm going to be doing is I'm going to be running around 50 field scale nitrogen rate trials around the state. This is predominantly corn and soybean, but these are going to be huge, I should say large scale, right? So we're using on-farm field scale equipment. It's 400-foot long plots, 90 feet wide, you know, using the spreader, getting good yield monitor data from all of these, and we're incorporating soil health into each of these, okay? That's predominantly focused on northern Missouri. And what we're really trying to get at with each of these is when we look at that nitrogen rate curve, right? We're all very used to seeing this response curve. We have, you know, as you increase nitrogen rate, at some point it plateaus and you can't get any more yield. So what these projects are really trying to look at is how does soil health shift this, okay? So for example, it could give you higher yields, right? So you get a higher yield, but your plateau is at a similar place. Another way that this game could go is that you could shift your AO&R. Oops, I hit too many times. But it could shift your curve so that you just hit agronomic optimum a little bit earlier on your fertilizer application rate. Or, hopefully, we could have both happen, right? You could have a lower AO&R and a higher yield. That's best case scenario. That's all kind of an open question. And so that's what this work is really starting to look at. And we're trying to do it, you know, on farm, and we're doing all kinds of variations with application of timing, form, using nitrification inhibitors, all that kind of stuff. So we're kind of making sure that the way that we're answering this is robust to how your nitrogen management program looks. Another one is that we're thinking about how to integrate soil health into a landscape. In Missouri, we have a lot of rolling hills, and so you can't manage your summit the same way that you manage the toe slope or the back slope. So this is a project that we've got going, and we're mainly focused here on yield stability, right? In a wet year, your toe slope, your gullies, you might not get much establishment and you get really low yields, right? Your summit might be a little bit better. And so how do we incorporate yield stability and kind of management and integrate soil health into that kind of thinking, right? So first one is this is a field that we're working on, and this is corn soybean rotation. And so we have 3 corn phases so far accounted for. So this is how we get our yield stability, right? Are we getting consistently high yields from the same places in the field? So we've got our 3 corn phases. This is some yield monitor data that we've got, and we use that to kind of group it into a couple different buckets. So we've got our yield stability zones, right? So this is moving towards that zone management idea. And so in the green here, those are our stable yields, and if it's dark green, that's very stable high yields. There's also such a thing as stable kind of lower yields. And then we've got unstable yields, which is in the red, right? And so generally unstable is a single bucket, and then the gray is, well, we're not quite sure. There might be other things going on. But so this is a project that we've got going on where we're layering in a couple different pieces of information. So we've got our yield stability zones, which is our outcome of interest, right? But again, we want to make sure that we are comparing things fairly within this field. So we have landscape data. So we have hill slope position, we have the aspect, which is which direction is it facing, right? North-facing slopes behave different than south-facing. And then we also have what's called a topographic wetness index. Generally, we just think about that as drainage. High topographic wetness index is where your field drains to, right? It's the low spots. And so we combine all those to give ourselves kind of these landscape zones, right? So those are the landscape level variables. The next thing that we want to think about is then, okay, we need to define our polygons. We have these overlapping things of yield stability, landscape, right? And we want to go out and take soil samples from the right spots. So we have about a couple hundred measurements here. We went out and did, this is like a 600, maybe, I can't remember, I think it's about 200 acres overall, this field. So we got, each of these polygons is about maybe 250 meters at the minimum that we sample from. And we went out and took a bunch of samples from each of these to get a representative sample. So again, we then combine those, and we are measuring our kind of standard soil fertility things that we're all familiar with, organic matter, extractable P and K, but then we're also layering on these soil health indicators to understand, hey, higher soil health, does that mean more yield stability? That's maybe a pretty important question when you're thinking about how to manage across an actual field, right? What we do is we then combine all of those, and we want to focus on that dynamic component, again, the part that is in your control and how it intersects and overlaps with your inherent properties, okay? So we're kind of layering all of those things together to look at yield stability. Now, yield stability isn't the only thing that you might be thinking about, right? But it does help us make some pretty important management decisions. How do you do like, your precision N application, your variable rate N? It also helps us think about things like seeding rate, right? And also then targeted conservation practices. You only have so much time to kind of get cover crops planted. You want to make sure you're getting the most bang for your buck, or you want to make sure you're not tilling the places that need to be no-tilled, or vice versa. So that's the type of thing where it helps us develop really specific sub-field management decisions. But, of course, this agronomic component isn't the, come on now, all right. So yeah, this is, we're now using this as well. We're doing some, at MU, we have our strip trials program, which is, we have about 20 on-farm trials, and I think we got about 15 of them to sign up for doing this. We're using this approach on-farm as well, right? And so we've got, again, big field scale. I think these plots on this are about 800 feet long, and they're about 120 feet wide. And we've got 5 reps with and without cover crops across each of them. So these are big fields, big scale, so we can get that landscape variability and understand how these soil health measurements can be used within a field to give targeted management. Okay, so we went in and did some pretty dense grid sampling that we're working on. But we're also interested within this same study thinking about pest pressure. In theory, a healthier soil means lower pest pressure. And for us in Missouri, we got a lot of soybeans, and so soybean cyst nematode is really important for us, right? It's a soil-borne pathogen, and in theory, again, some of these soil health indicators should relate to lower pest pressure. So when we've been going out and looking at those sites, each of them, we've also been retaining part of that soil sample to do a soybean cyst nematode analysis. So we're working with our IPM that we've got at University of Missouri to understand, again, how all of these things combine to make both agronomics and pest pressure kind of come together, right? And again, this helps us with things like selecting varieties, right? I'm not sure how much soybean cyst nematode is a problem here, but in Missouri it's a big problem, and kind of the only thing we got is selecting a resistant variety And so that's kind of helps us do varietal selection. If you are, for example, if the only part of your field that has soybean cyst nematode pressure is your stable, low yielding part, you might not be as concerned, right? And you might not have to switch to a resistant variety quite yet. So it's things like that that we want to start walking our way through and developing kind of decision-making processes. Oh, again, yeah, okay. And all of that is because we want to use soil health assessment to get you to the outcomes that you want, right? Then those outcomes might be varied, whether it's yield stability, thinking about your nitrogen management program, or thinking about, varietal selection, seeding rates, etc. Because I like to think about soil health indicators, they're supposed to be decision-making tools for you. And so if you can't use them, that means I'm not doing my job right. Because again, this is this management connecting to outcomes. So this is my shameless plug for the Soil Health Assessment Center that I run, where we're developing these tools, and we can run a lot of these assays. They're not commonly available in commercial labs yet, but this is part of what we're doing. And we really focus on kind of two efforts here. So the first one is that we want to make sure that these indicators are robust in space and in time. Does it matter? How much does sampling timing matter? How much does your specific context matter, right? We want to make sure that it's robust across different systems, right? If you're doing corn, soybean versus a wheat rotation, or if we're down in the Missouri Delta where we've got cotton peanuts is a really common one. And we are really dedicated to doing this in that on-farm conditions, okay? Next thing is we want to make sure that those indicators not only have we sufficiently kicked the tires on them, but they actually relate to the things that you want. Again, this is your diagnostic tools, and we want to think beyond just total yield, right? We want to think about things like efficiency, yield stability, pest pressure, nutrient use efficiency, etc. So with that, I want to say thanks for listening. It was a pleasure to be here, and I now have some time for comments, questions, etc.
Questions for Dr. Whitaker?
Yep, sure.
I'm just curious if you're looking at incorporating livestock.
So some of them did have livestock. We have a project going on right now where we're looking at grazing cover crops. Missouri's got a lot of beef, especially because we've got some really bad hilly ground. And so we're thinking about incorporating livestock. The problem so far on whenever we have research plots or strip trials is that it's hard to control livestock. I think the best we've done so far is incorporating manure. When I talk to my colleagues like about silvopasture or something like that, the spaces you need get real big, real fast, and the number of head of cattle you need and rotating them gets pretty intense. I'd like to start investigating that a little bit more, but the logistics of it are kind of complicated. You might have better solutions down here than we have in Missouri. But so in terms of we have applied manure, but not necessarily grazing and that kind of direct manure depositions.
Any other questions? George, thank you very much for making.
Thank you.